We're going to transition from surveys and sampling into experiments. And one of the biggest things to realize at the beginning is to know whether or not you're doing an experiment. So read this article. A lot of times places publish results and we automatically go to a cause and effect, and not always is that the case. So read the article, pause the video, and then unpause it and we'll keep talking. So, based on this study, should we conclude that smoking during pregnancy causes children to have a greater chance of developing ADHD? Okay, we read this article and we automatically say, oh, smoking causes ADHD. Not necessarily true. We cannot say that. We should not conclude that. Okay? The reason is this big word causes. We can say that yes, there seems to be an association, but the big thing is that unless you're doing an experiment, and a very well designed experiment, you cannot say that you have a causation relationship. This is not an experiment. Okay? All I'm doing is surveying. Okay? This is what we call an observational study. Whenever I survey or just observe, it's an observational study. And with those, I cannot say that I have any type of causation. So from this, I can assume that there's an association, but I cannot say that there is a causation. And a big thing to know for AP, that just because there is an association, that does not imply causation. Association does not imply causation. Okay, so let's get specific about some differences between observational studies and experiments. An observational study is just that. All you do is observe. You may sample randomly and then either survey or just observe and measure results. In an experiment, we are going to apply some sort of treatment. Know that it is an experiment if you can directly affect one of the variables. Okay? In the article above, there was no affecting anything. I did not make anybody smoke. I did not make anyone have ADHD. In an experiment, you have a direct effect on some variable. Okay? That being said, we need to watch out for lurking variables. Lurking variables are more common in observational studies, but they can occur in experiments if you don't design it properly. They are variables that you cannot account for or you do not account for that could cause some kind of problem. Okay? A lurking variable could be the fact that mothers who smoke do not seek prenatal care as thoroughly as ones who do not. Okay? There could be some other variable that you are not considering that is causing this problem, and that is a lurking variable. Okay? Confounding is when the actual variable, the lurking variable, causes the problems. You get two variables that associate in such a way that you can't tell which one's causing which. Two variables associate and you can't tell which one is causing the effects. Okay, so here's the thing. AP tip time. A lot of people have a problem distinguishing between the two. Okay, every lurking variable is a potential confounding variable, but it does not have to be. Some variables could cause problems. They might be ones that you are not controlling for, but they don't necessarily. Okay, so lurking variables can be confounding or can cause confounding, but they don't always have to. Okay, so let's look at this example and we'll see what it tells us. The November 2009 issue of Nutrition Action discusses what the current research tells us about the supposed benefits of soy. Okay, for a long time we thought that soy explained lower rates of breast cancer and other diseases. So they conducted experiments, and soy either had no effect or had a very small effect on the health of participants. So, for example, they did studies on randomly assigned elderly women, and they either gave them soy or placebo, and none of the studies of soy showed that it was more beneficial for preventing osteoporosis. So, what explains the difference? Okay, we don't know. But there must be some lurking variable, or some, which is now a confounding variable, that we are unaware of. There is some variable that explains this difference since soy does not. So it could be genetics. Genetics could be a lurking variable. Other dietary factors other than the use of soy, or any other difference between the two cultures. Okay? There are variables that we are unaware of that are causing this relationship, since we're showing in the experiments that soy is not causing it. Okay, another AP tip. Don't just rely on the vocabulary, because there's a chance that you might use it wrong, and they don't want you to just spit out vocabulary. It is really important to know the vocabulary. However, it's more important to explain what you mean. So instead of just saying that blank is a confounding variable, okay, explain what you mean by confounding variable. Pause the video, look at these check your understanding questions, and then start the video again and see if you got them right. The first one, number 1, is an experiment. You have a direct effect on one of the variables, the brightness of the screen. Some of the screens were turned down to a low brightness, some of them were turned up to a high brightness, but that was a variable that you have a direct effect on, which means you're applying a treatment, which means it's an experiment. Number 2 is an observational study. Okay, there are no treatments being applied. We are just observing with a survey teenagers who eat with their families and their GPA. So the explanatory and response variables. Okay, they're just what they sound like. An explanatory variable is the variable you're looking to explain, okay, the relationship. So the explanatory variable would be the number of times per week teenagers eat with family. The response variable is where you're looking to see the effect, and that's in GPA. Okay, and number 4 is an observational study. So that is why there cannot be a cause and effect. So there may be a lurking variable that we are unaware of that's explaining this association. It might not be the fact that they eat with their families. Maybe it's that parents who eat with their children are more involved in their kids' studies. Okay, let's get into the actual vocabulary and language of experiments, and some words that we need to know. 1, a treatment is the specific condition that is applied. Usually it has to do with what I'm wanting to know about. So if I'm wanting to know about the relationship between the brightness of a computer screen and the battery life, then the treatment will be the different brightness levels of the computer screen. Okay, the experimental units are the smallest collections of individuals to which the treatment is applied. Another AP tip, this is a common error on the test. Many people don't pay close enough attention to the problem to realize that an experimental unit may be something other than just one person. So watch and read carefully. Okay, subjects are just experimental units that are humans. We call them experimental units unless they are humans, and then we call them subjects. Just sounds a little more humane. Okay, factor is also what we call our explanatory variable. It's the variable that we're changing. And level is the specific value of a treatment. You can use the factors to help you figure out how many levels there are. If there is only one factor, then however factors there are, the factors equals the level. So if there's only one factor, there's only one level. Okay, if there are multiple factors, there are going to be multiple levels, and it's going to be depending on like combination of things, and you'll see that in a second, one of our examples. But these are all 5 vocabulary terms that you need to know. Okay, so let's look at this study and see if we can pick out the different things. So a study published in the New England Journal of Medicine compared 2 medicines to treat head lice. Okay, so we have 2 medicines. 812 people and 376 households in 7 areas around the world. Of 185 households randomly assigned one medicine, so 171 of them were free from head lice. Only 151 of the 191 randomly assigned to the other. Okay, so we want to identify the experimental units. Most people would say it's the individuals, but it's actually the households. Okay? We are randomly assigning medicines to households, not individual people. You are not going to get 2 people in the same house on different medicines. So this is households. Okay, the explanatory variable, that's going to be the medicine. The explanatory variable is the medicine that they were assigned. The response variable is, did they get rid of lice? Yes or no? Okay, and the treatments, okay, were just the 2 types of medicine. So notice that since I only have one explanatory variable, then the factors are just the explanatory variables. One medicine and the other medicine. Let's look at one more example. It's at the top of the next notes page about these different vocabulary words. So I want you to pause the video, read the example, see if you can pick out the different things, and then see if you get what I get. Okay, so identify the factors for this. The factors are going to be whether or not the fertilizer was applied and the amount of water. We have 2 different things we're looking at to explain our growth or our response variable. Okay, since I have 2 different factors here, yes or no, fertilizer or not, and I have 3 different factors here, that means that there are 6 different treatments. There's 2 levels of this, 3 levels of this, so 6 levels overall, 6 different treatments. Those treatments would be fertilizer with a half a gallon of water, fertilizer with 1 gallon of water, and fertilizer with 1.5 gallons of water. Then there would be no fertilizer with a half a gallon of water, 1 gallon of water, 1.5 gallons of water. Those are all the treatments. Okay, so now the final question is, what is the experimental unit? And in this case, it's going to be the tomato plants. Okay, so hopefully you understand those 5 vocabulary words. If you don't, make sure that you ask about them in class because it's very important that you understand. Okay, so now let's talk about, we've talked about the vocabulary words. Let's talk about what bad experiments look like. Okay, I have this idea, and let's say we wanted to design an experiment to see if caffeine affects pulse rates. So we have an initial plan. We're going to measure the pulse rate. We're going to give everyone some caffeine. We're going to wait for a specified time. We're going to measure final pulse rates, and we're going to compare the final initial ones. Okay, some problems. One, some people might be anticipating it. They might be saying, I'm going to get caffeine and it's going to make my pulse rate go up, and since they're expecting it, it happens. Okay? That's actually called the placebo effect, but we'll talk more about that later. We're not saying what happens while we wait. Okay? What if I tell a joke and some people laugh? Or how do you know that the joke and the laughing is not what increased our pulse rate? Depending on what we drink, if we're drinking Coke, what if it's sugar that's actually affecting the pulse rate? Okay, that could be a lurking variable. So sugar. Also, how long we wait. That might be a lurking variable. Okay, if we wait too long, maybe the caffeine runs, heart rate goes up and then comes back down. If we don't wait long enough, maybe it doesn't go up as high. Your body size is a lurking variable. Some people may affect, caffeine may affect some people more than others. That's a lurking variable. Whether or not you've eaten, so if you have food in your stomach, if you've had caffeine recently, that might be a lurking variable. All of these things are things that this experiment right now does not control for. But what we need is a way to compare. Comparison solves a lot of problems with lurking variables and helps them keep from turning into confounding variables. Comparison keeps us from having confounding. So here's what we do. One, we need a control group, a group that is not going to eat or not going to drink any caffeine or intake any caffeine. We need to randomly assign people to that group or the caffeine group without them knowing which group they're in. That takes care of that anticipation, like getting excited because you're expecting the caffeine. If it happens to one group, it would happen to the other. Okay? We want to make sure everyone's drinking the same amount of caffeine at the same rate. And we want to make sure we randomly put people in one of the two groups. That helps us even out any lurking variables like body size and caffeine tolerance that we just can't control for directly. So, and I really and truly what we want to do and how to fix bad experiments is to make sure that you have two groups, a control group that doesn't intake any caffeine, in this case, and then the caffeine group. They don't need to know what group they're in, and we need to help try and make sure the groups are as similar as possible. That way we know that any differences between them, any differences in heart rate is attributed to the caffeine and not something else that happened. Okay, so we will talk more about this in class, and we will actually go through more of this experiment in class. But in general, this is what a bad experiment looks like when you don't have any comparison. Yes, we're comparing final and initial, but we need to be able to compare that increase or decrease from one person to another. So you need comparison. That is what is most important in experiment design.